Hello and welcome to the second lecture of Applied Environmental Microbiology. Today we are going to talk about the history of environmental microbiology, how microbiology first developed, and we moved from very generic sanitation engineers to environmental microbiologists. So let's take a look. Initially, when we were trying to understand what microbiology is, the scientists and the engineers together were stuck by immense diversity that is present in the environment. It was very difficult to tell microbes apart from each other because we did not have the right technology to do it. But as technology improved, developed, we could start distinguishing between different kinds of microorganisms, their activities, start even making trees like this one, for example, where the orange is bacteria, the green is eukaryotes, and the one in middle is archaea, and start understanding how each of these branches on these trees are very unique microbes that have very different activities and behavior in environment. The latest technology to do this is called sequencing, and I would like to take a moment to explain to you that one of the intentions of this course is to acquaint you with the latest technologies that are accessible and available to you in India that can be used to understand environmental microbiology better. So we'll be focusing more on what is the current microbiology, but first we need to understand what the history of microbiology is. So as I said before, scientists were in awe that the microbes, or in other words smaller forms of life, were present in very diverse and very different environments. And this table here shows to you some extreme environments in which we have seen microbes not only surviving but also thriving. For example, hyperthermophile microbes, very much thermophile, who love high temperatures, have existed in undersea volcanoes where minimum temperature is 90 C and maximum temperature can be boiling hot and more, 122 Celsius. So these microbes actually survive and love this immense heat. And this is very interesting story about how archaea, a very different domain in microbiology, was discovered in these undersea volcanoes, and we'll talk about that story later. But imagine scalding hot water, they'd love it. And then we have extreme low temperature psychrophiles, one who love low temperatures. They have been found in deep sea ice, where temperatures range from -12 degrees Celsius to 10 degrees Celsius, and bacteria love living in this cold temperature. So if you see some psychrophiles growing in your refrigerator, don't be alarmed, they love it. Microbes have also been found in acidic sediments, acidic hot sediments, where the pH drops below 0 and rarely rises above 4. These are acidophilic microbes. Acid, phile means loving. Then we have alkaliphiles, the ones that love alkaline pH, and these have been found in soda lakes where the pH ranges from 8.5 to 12 microbes. When it comes to extreme pressure, where will you find extreme pressure? Deep ocean, obviously. So in deep ocean, where the pressure ranges from 500 to more than 1000 atmosphere, microbes are known to thrive. And thus we know that we have barophiles. Similarly, we have microbes that love and grow in extreme salty environment, and they are found in salterns where salinity can range from 15% to 32%. Important to note that 32% is often the maximum salinity we can achieve on our earth, the surface of our earth. Note here, out of these extremophiles listed here, only 2 categories are bacteria, rest are archaea. And when we talk about microbes, we often talk about bacteria, we talk about virus and fungus and maybe protozoa, but archaea, yeah, let's explore them. One of the first discoveries in microbiology was when we started poking into our intestines and used some wonderful microscopies to see what's going on in our gut, and we found that our gut was colonized by as much as 10 to power 11 microbes per gram of biomass in our gut. And we made these beautiful pictures. Again, this is an artist's impression, and we found out that these microbes affect us, but we didn't know what kind of microbes are there. What we started finding out soon was that we can study microbes on individual level, population level, and community level. Initially, it was just easy to treat microbes as a black box. You have bad microbes in your gut, take an antibiotic. We need microbes to treat wastewater treatment. Everything that is left is called sludge, that's biomass, without distinguishing different kinds of microbes. When we developed culturing techniques, which is that we can grow those microbes in our lab. So we take microbes from this picture, from our gut. This is gut microbiome, by the way. We take these microbes, let's say the blue rods, and we grow them on plates. So once we have grown them on plates, we can study them individually. We can see how they express different proteins, how they are built inside, what their genetic code is, how do they behave under different environment. Then what we can also do is we can put multiple microbes together and we can enrich them for a particular purpose. This is when we study them on population level. So this would imply we want to know, okay, we have a mixed community or we have many microbes of the same type. What is their growth rate? And this is some environmental engineers must be remembering about monokinetics and kinetics of growth. And don't worry if you don't, we'll cover all of that again. But on population level, we study demographics of microbes, and this is very important for environmental engineers, where we are interested in the right food to mass ratio. We want to know exactly how much acetate to inject in our groundwater to sequester uranium, right? And then on population level, we are also now recently interested in population genomics, where we are trying to understand how population of microbes varies when the environment varies. For example, let's say earlier we had clean groundwater, so we had a particular microbiome in the groundwater, and then fluoride and arsenic levels increased exponentially, let's say. How did the microbial community respond to it? And this is very important thing to understand. And this is important because this is exactly where we understand and we know that we are likely to find out microbes that are very unique. For example, microbes that sequester chromium out of water, microbes that can survive up to 3,500 ppm of chromium. We also look at population genetics to see how microbes of a similar kind, let's say I'm interested in cellulose degradation, let's say I work with cow dung and I'm very interested in seeing how to degrade the cellulose. Now there is a family of microbes very famous, Clostridia. Many of the cellulose degraders are known to belong to Clostridium. I want to see how within Clostridia the genes that are responsible, that help microbes to degrade cellulose, how they vary. So this is allele frequency distribution. We'll be talking about this as well. And now because we have a tool called metagenomics. Metagenomics is a promising, wonderful tool that has been recently developed. Meta means big, genomics means study of genes. So here in metagenomics, what we can do is we can sequence millions and millions of microbes, either only a targeted gene that we are interested in, or we can sequence the entire genome from as many microbes as possible to get billions of sequences. So in one go, we can generate tremendous amount of data to know what different kinds of microbes are present in the community that I'm studying, what are their functions, because now I can even go up to genome level and find out what genes they have, what genes are getting enriched. Metagenomics is a big departure from the earlier microbiological techniques in the sense that not only is it wonderfully fast, rapid, but also very cost effective. And it won't be an exaggeration to say that we are on verge of my metagenomic revolution. In India, I have seen that metagenomics is often referred to just amplifying high throughput amplification of a particular gene and sequencing it. Let's say I'm interested in gene A because gene A is related to methane formation, methanogenesis, MCRA, that's the gene name by the way. So I'm interested in gene A, which is MCRA, and it's responsible for methane formation in anaerobic environment. And what I can do is I can take my environmental sample. Let's say I have a drill bit that goes into a petroleum oil field, oil well, and I get some sample from there, and I want to know how much methanogenesis is happening down in the well. So I can extract the DNA of the microbes there very easily, all of them, billions and billions of microbes. I can extract the DNA. Note, not all billion microbes are same, they're very distinct. So if you have billions of microbes, you might have at least some million different kinds of microbes. So I extract all of their DNA, and then I can amplify or increase the concentration of the gene that I am interested in, gene A or MCRA, and I can sequence only that gene. And now you might ask, if I already know the sequence of gene, then why is it important to sequence it? Because in microbes, in environment, we notice that the same gene might have different, may have some diversity. Some ATGC, some codes might be different, might vary, and this is what is referred in India as metagenomics, where I amplify one gene and see how it varies in the community. And the most popular gene for this kind of metagenomics is 16S rRNA, which gives me information about what the microbe is. In other words, what name can I give to the microbe, not what it is doing. The example I shared with you earlier tells me what it is doing. This microbe, because they have MCRA gene, is generating methane gas. But 16S rRNA will just tell me what's the name of microbe, and if I have the right knowledge, I might be like, oh, I know this microbe, it creates methane, or this microbe eats cellulose, but it's not giving me functional information. Outside, in many Western countries, this kind of sequencing is called as high throughput amplicon sequencing, and metagenomics refers to whole sample metagenomics, which is, I'm not only interested in a gene, I'm interested in every single genetic element in the microbial community. This is where the community genetics come into picture. As I promised, let's dig into the historical roots of microbiology. Believe it or not, some centuries ago, not a long time ago, some centuries ago, the general level of hygiene in Europe, the much developed Europe, was pitiably worse. People rarely wash themselves because they do not understand that not washing them will make them sick. And it was not until when Robert Hooke invented a better microscope that he actually could see, oh look, these are microorganisms, these are small little microbes or microbe kind of things in mold, in fungus. And it's important to note that fungus, the microbe fungus, is much bigger than a bacteria. So fungal cell is much bigger than bacteria, it's easier to see. So Robert Hooke was the first person who actually made a good microscope and used it well. The next gentleman who came into this picture was Antony van Leeuwenhoek. I don't know how to pronounce that name, but that's the best I can come up with. He invented a very simple microscope, and he reported pictures of V animalcules. So V means really small, animalcules like molecules, like really small animals. And he was the one that actually popularized and showed that these V animalcules, now we know microbes, are found even in drinking water. So you can imagine the shock of an elegant English woman realizing that the water she's holding in her hand and drinking is full of microbes. But don't be alarmed, even though our drinking water has tons of microbes, most of them are benign, they won't hurt us. So drink water. And then science developed further. Scientists like Kohn, they developed measures to prevent microbial contamination in lab. And this is where Louis Pasteur came into picture, the scientist whose quotation I quoted earlier in previous lecture. So Louis Pasteur, Kohn, they developed techniques which allowed us to preserve food longer, to make sure that our milk does not go bad. This also led to medical understanding why we should disinfect our wounds and our hands every time they get dirty. Now let's take a moment and wonder, what kept scientists from developing microbiology before contribution of Hooke and Leeuwenhoek? So remember the V animalcules of Leeuwenhoek and the simple microscope of Hooke. So maybe if you're watching this video, pause it for a moment and think about it. Why didn't microbiology develop before these two phenomenal scientists came into the picture? And yes, perhaps you have guessed it right, that we need the technology to see such small microbes that are definitely invisible to our naked eyes, to make sense out of them, to understand what they're doing. We didn't even know a life existed beyond what we could see. And now, as I shared in the previous lecture, we know that most of the life forms are not what we can see. Most of the life on earth is actually the nearly invisible to human eye microbial world. And thus, and next question that I want you to think about is, was invention of microscope the final frontier in microbiology? I can loosely rephrase this question and say that we had discovered microscope back then in time of Hooke and Leeuwenhoek, the French scientist. Did we do everything that microbiology had to do? Have we discovered everything we need to know? Do we know about all the kinds of microbes that are present on the earth, or is there any more scope for research? Are there frontiers that lie unexplored? So again, I suggest pause the video for a few seconds and think about it. And exactly as you might have noticed and hopefully guessed, that no, invention of microscope is not the final frontier in microbiology. Every year we are developing new techniques and we are learning more and more about microbes around us. Now, how did this all begin? It began with ancient scientists, Antony von Hooke and Louis Pasteur. And Louis Pasteur was the first who demonstrated that spontaneous regeneration theory is wrong. Now, what is theory of spontaneous regeneration? Earlier, when people did not understand microbes, but thanks to Hooke and Leeuwenhoek, we knew that these microbes, these tiny animalcules, actually exist all around us. People believe that they spontaneously regenerate. This is a fancy way of saying that, well, first there were no microbes and then suddenly there were a lot of microbes. This is, oh, the food was clean. Let's say the chapati or the dosa was clean, and few days later it fermented or fungus grew over it. This was spontaneous regeneration. I did not go and put a fungus there, because to human eye and mind, for every plant, for every living being, there has to be a progenitor, and the progenitor for the microbes was not visible. So people believe that they spontaneously regenerate, and this is also in line with the creationist theory. We are not going to talk about that in this course though. So Louis Pasteur was the first one to demonstrate that certain chemicals are catalyzed by microbes and spontaneous regeneration is not correct. And when he understood that spontaneous regeneration is not right, only then he could develop methods to stop food from going bad, from sterilizing. And here's a cool thing, we still use his techniques in lab to make sure that our flasks are sterile. We still use his techniques to preserve our food for longer times. So Louis Pasteur discovered two important things. First, microbes are selective in what chemicals they interact with and how they interact. So in the sense that they are not just random things growing there, they have some program behind them, or in some intention behind them. The other thing he proved was that they don't spontaneously regrow. For a fungal growth to happen over a chapati or over dosa, we need some fungal spores or fungal microbes already in the air or in the environment. And the way he did this was by modifying the existing laboratory setup to make a beautiful flask with a neck that bent in a unique shape. So let's take a look at the slide and see here. This is his this is a cartoon of his experimental setup where he had broth. Broth is food for microbes. They love this food, very carbon rich, nutrition rich. And then in his glass where he got this special bend made. So with this bend, he prevented microbes from entering into the broth. And first he boiled it so that all existing microbes would die. Then he cooled it and he let it sit for a week, 2 weeks, 3 weeks, and it did not get contaminated. So this suggested that, oh, there is a possibility that microbes will not spontaneously regenerate or generate. But this does not prove that they do not spontaneously grow. This just shows that in certain circumstances they don't spontaneously grow. So what he did next was he removed the curved neck. He removed this neck and he let it open so the air could freely move in and out. And he noticed that microorganisms grew in the broth and it became cloudy, suggesting that if exposure of air is allowed, then microbes will regrow and will grow. So this says that for any life to form, there has to be a progenitor. If you can't see, it doesn't mean it is not there. The next great scientist in microbiology was Koch, and this is his work on infectious diseases. He came up with the idea of germ theory. At this time, people did not understand that there are infectious agents that we can call germs, which are microbes, and they are the ones that cause diseases. There were some really backward, rudimentary, and now senseless. Now we believe they are senseless ideologies about what diseases and what medicines are all about and what they should be. So Koch was the first one to come with the germ theory, and he gave wonderful 3 postulates that we'll talk about very soon. Now, what were these postulates and why did he give them? He wanted to associate the factors, the disease agents, with the cause of disease. And let's look at this, his postulates, to understand the logic of Koch. In order to identify disease agent, first we must find it in all sick individuals. So this is what this wonderful scientist did. He took a particular disease, disease A, and he took sick mice that had the disease A, took out their blood and looked at it under microscope to find what is this unique thing that's present in all sick animals and it is not present in healthy animals. So first Koch postulate is that the disease agent must be present in all sick cases and must be absent in healthy. But this does not prove that the disease agent or the unique agent that he has identified is actually the cause of the sickness. It just says, well, in a sick patient, in a sick mouse, we will find the diseased agent but not in the healthy one. So the next thing he suggested that we need to do to prove that the disease agent is actually the disease agent was that we need to isolate the agent and then grow it in vitro. So what he suggests that we need to take the agent from the sick microbe and then grow it in our lab. Once we have grown it in our lab, we need to inject it in a healthy host and make sure that it ensures disease. So in other words, there are 3 steps to Koch postulates and to prove that a particular agent is a disease agent. Now remember, this is the time of microbiology when we did not have germ theory, we did not know why and how people fall sick. So let's say there's a disease A and there are mice that are infected with disease A. So he collected all of them, as I said before, he did their blood analysis, looked at it under microscope and found out that there are certain animalcules, for lack of other word, that are unique and only present in sick animals, not in healthy animals. This next step of Koch postulate is that we should remove these agents, these unique agents that are present only in sick animals, and grow them in lab. Once we have grown them in lab, inject them to healthy animals and wait and watch. If healthy animals fall sick, we can say that this particular thing that we had grown in lab and separated from sick animals is actually the cause of disease, is what we can call as germ, the disease agent, and hence the germ theory was given. And once he did this for the first time, he went on and did it for tuberculosis, and these postulates are still used when we try to identify the agent for a particular disease. A wonderful example would be SARS epidemic in China many years ago. So many years ago, a new disease broke out in China, a severe acute respiratory syndrome. We sort of knew that it is viral by looking at its characteristics and the quick blood work, but we didn't know what virus it is. And at any given time, there are multiple viruses that are present in human body. So the scientists actually followed a slightly modified version of Koch postulates to separate and isolate the infectious agent. And this is a beautiful diagram showing how Koch did it. First, he isolated the microbes from a sick or dead animal. He grew them in pure culture. He injected it in a healthy mouse, and the healthy mouse also started falling sick. And then he saw that the agent is also present in this sick mouse now. It was not present earlier, but it's present now. And he could grow it in the lab, and both were same. So it is this agent that makes a healthy mouse fall sick. In modern era, right now, we do not need to rely on these multiple steps of colony formation, looking under microscope, and maybe very slowly and very painfully characterizing the microbes and seeing what kills them, what doesn't. We can directly go and use some wonderful techniques. It all started with Sanger. Sanger, a scientist, he invented this technique of Sanger sequencing, wherein we could actually decode the genetic code.
So the beauty of Sanger sequencing is that it does sequencing by the method of synthesis and not by the method of removing the nucleotide, which were what people had earlier tried and failed. So in Sanger sequencing, let's say you have a sequence here, and each of these gray blocks are your unknown nucleotides, and what happens is, instead of using the generic nucleotide that we have, adenine, guanine, cytosine, thymine, what in Sanger sequencing we do is we use the didoxy format of the nucleotide. So what happens in the first cycle, the machine will allow A, TGC, dideoxynucleotides to come and replicate, to come and start replicating. Now as the replication happens, when the first correct complementary match will attach here, so we can see here it says A has attached successfully, A dideoxynucleotide. It means that the complement T was present in the genome. So when the A has attached here, then the replication cycle breaks, because didoxy, due to its very nature, will not allow the replication cycle to continue. So for the next replication cycle, now we'll have the replication will end at A, and then the next gray block here, the unknown nucleotide, will again be exposed to different kinds of dideoxynucleotides, and the one that attaches here, we know which one is attaching, because we don't send them all together, we send them one by one, and once C has attached here, we know that G is present here, and so on and so forth, until in each replication cycle we get to know base by base what nucleotide is present in what sequence. And as a result, Sanger sequencing has very low error rate, but it is also time consuming, and even though the costs have dropped a lot, in comparison to other sequencing methods, it still remains to be very expensive.
So earlier, when we were still struggling with sequencing, we knew about bacteria, we knew about virus, we knew about eukaryotes, so we knew something about protozoa, about fungus. There was a scientist, German scientist, Carl Woese. Now Carl Woese, he had this passion to find out about life in extreme environments. So he would find himself in places that no other scientist would be willing to go. So he would go to the hot springs that are on average at a temperature like 90 degree, minimum 80 degree, 90 degree, 95 degree, 100 degree even, boiling volcanic streams, and he would collect samples there. And you can imagine stepping into really boiling water is not something anyone would like to do, so he definitely had a safety suit with him. He and his students would take samples from there and would try to reproduce these conditions in lab. And one might wonder, why would he do that, and who would fund such research? And the answer is very simple. Carl Woese was trying to understand how life developed when Earth was still in infancy. So when Earth was in infancy, it was hot, sulfur-rich, anaerobic environment, definitely not the pleasant Earth we know now. So he would find these places on Earth that are still sulfur-rich, are hot, and have extreme pH, extreme temperature, and see if there is any life. That's what he wanted to know. So when he would take samples from there and try to regrow the life forms, or to detect life forms in those, he found there is life. So he knew that, okay, life can start here. We knew it logically, but now we know it experimentally. And then using these preliminary techniques of sequencing, he actually sequenced the genomes of certain organisms that he could grow at extreme high temperature in sulfur-rich conditions. And the funny thing he found was that these genomes looked very distinct, very different from the genomes of bacteria. So he knew that perhaps they are not bacteria, or maybe they are different kind of bacteria. There was some confusion there. So what he did was he tried culturing these organisms so he could see if their morphology was same or not. And culturing them is an immensely difficult task because they need anaerobic, extremely hot and unique environment, which is hard to create. But finally, after many years of attempting this, he succeeded. And what he noticed was that not only are their genes very different from bacteria, their inner structure of the cell is also very different. Many features they actually share with eukaryotes, and some features they share with bacteria. So they are neither bacteria nor are they eukaryotic, the new cells, the cells with nucleus. There is something in between them, but not quite between them, because they are very unique in the way their cell membranes are made. And then he started asserting in the scientific community that this is a different domain of life, this is a different kingdom of life, they should be given a separate name, and he chose the name Archaea. So, but I call in this slide, if you notice, I say Carl Woese, brave pioneers. Carl Woese and other scientists were condemned for even thinking that there is a life form beyond bacteria. And that's why, and they struggled a lot to make the point, and eventually it emerged true that yes, now we have bacteria, archaea, and eukaryote, 3 different forms of life. But now scientists don't have to struggle so much, because now is the era of genomics and genetic engineering. Fancy words, genomics. Genomics is when we can actually look at the entire genes, know what they are, quickly match them with existing database of genes of microbes that we know, and sort of guess what actions they do, what their qualities would be. Genetic engineering is when we can actually engineer the genes to suit our purpose. This is something that biotechnology works with. Recombinant techniques and genetic engineering are definitely in domain of biotechnology. We'll go very briefly over them, but not in depth. And now we have CRISPR-Cas. CRISPR-Cas is a promising technique that actually, so genetic engineering and recombinant techniques have lot of limitations. We engineer genes in vitro, not in vivo. What it implies is that we engineer genes outside a living cell, and then we put it inside an living cell and we let it multiply within that community, and then we extract the gene or the protein that we are interested in. But CRISPR-Cas, what CRISPR-Cas can do is we can inject and we can operate, we can engineer the genes of a living cell, and this is very cost effective, very accessible, and promises treatment for many medical problems such as antibiotic resistance. So we can actually go inside a cell, bacterial cell that's antibiotic resistant, tweak it so that it stops being resistant. It's also promising for cancer research, CRISPR-Cas, that we can, when cancer cells stop responding to treatment, we can modify the cells so that they start behaving better and respond to treatment better. This is Carl Woese, by the way, just paying homage to a wonderful scientist, and these are the preliminary sequencing sheets he had, and these are very long, very painstaking work to do, and this is the result of his wonderful work. Now we have a new kingdom, archaea, and as I mentioned, this is era of genomics. Now this is Illumina platform. They are one of the leaders in high throughput sequencing, and I'll go over this later in detail, how this works and why it's useful and where it is useful, where it is not useful. For now, I think this is a good time to revise this lecture and the introduction we had about Earth being a microbial planet. So this is where we started our lecture series from, trying to understand that what we see is not all that there is to Earth. There's more to life, there's more to our planet than the big life forms that we see and notice, and most of it is actually microbial. And we also noticed that the microbes existed long before us, long before us, and will probably outlive all higher order of life forms, because remember we talked about microbes that can survive and thrive in extreme environments. So if there is a catastrophe and higher orders of life are wiped out, and let's be very clear, we are already in middle of an extinction phase on Earth, so species are going extinct very fast right now, mass extinction, but microbes will probably outlive, not necessarily in the way they're living right now. They'll evolve because they evolve really fast, but they will. It's also important to remember that microbes are a majority in our human bodies. We have nearly 10 microbial cells for every 1 human cell. They're very important for our health, and very few of them actually hurt us. Most of them help us. And cyanobacteria, the green bacteria we see on our lakes, is often the bacteria that made Earth's environment aerobic. So we really need to express and feel gratitude towards these tiny organisms whose microscopic picture is on the left side, that have allowed life forms such as ours to evolve on Earth. And let's spend a minute here to look at this left side of this slide. We notice that we can see here that we see strands much like hair. This is not one cyanobacteria. If you look very carefully, you can see these small compartments. Each of these small compartments is one cell, and they like to connect to each other in hair-like structures. Then we also learned that Earth is a microbial planet, and the infinitely small might be playing an infinitely large role. This is all for now. Thank you. and I'll see you in the next lecture. Thank you.